Hello everyone, we're continuing our training. The previous section explored human-created biodiversity. Breeding methods were also considered, the hybridization, mutagenesis, and genetic engineering. Today we are starting a new section about systematics of living organisms. And today we have the Zem number 4 about prokaryotes and eukaryotes, real bacteria and their classification, Archaebacteria and their classification and features, viruses, their classification, and ecology. Organisms whose cells have a nucleus are called eukaryotes. Organisms whose cells do not have a nucleus are called prokaryotes. Both types contain DNA as genetic material, have a surrounding cell membrane and cytoplasm, have ribosomes, accomplish similar functions, and are very diverse. And distinctive characteristic of prokaryotes about this. First, most of these prokaryotic cells are small. Second, DNA is located within the cytoplasm. Third, prokaryotes rarely have any membrane-bound organelles. And last, prokaryotes include bacteria and cyanophytes. About distinctive characteristic of eukaryotes. First, eukaryotic cells are much larger than prokaryotic cells. Second, DNA is found at the nucleus. Third, eukaryotes have membrane-bound organelles, and last, eukaryotes include plant and animal cells. Bacteria are microscopic, single-celled organisms that thrive in diverse environment. Their organisms can live in soil, the ocean, and inside the human gut. About characteristics of bacteria. Bacteria share a number of common structures that are briefly described. First, slime or extracellular polysaccharide. This is extracellular material loosely associated with the bacteria that is elaborated by some bacterial species that facilitated colonization of smooth, prophylactic surfaces such as intravascular cavities. Second, capsule. This polysaccharide, other cation of the bacterial surface, often plays a role in preventing phagocytosis of bacteria. Third, Peptidoglycan or cell wall. This provides bacterial shape and rigidity. The cell wall consists of alternating units of N-acetyl-glucosamine and N-acetyl-muracil acid. The polysaccharide chains are cross-linked by a peptide bridge. It is a primary target of antimicrobial therapy because it is specific to prokaryotes. About reproduction. Most bacteria multiply by a process called binary fission. In this process, a single bacterial cell, called the parent, makes a copy of its DNA and grows larger by doubling its cellular contents. Some bacterial species, such as cyanobacteria and firmicutes, reproduce via budding. In this case, the daughter cell grows an offshoot of the parent. And about classification. A few different criteria are used to classify bacteria. The organisms can be distinguished by the nature of their cell walls, by their shape, or differences in the genetic makeup. There are three basic bacterial shapes. The first, round bacteria called cocci, and the cylindrical, capsule and shaped ones known as bacilli, and spiral bacteria ently called spirilla. These shapes and configuration of bacteria are often reflected in their names. However, the most recognized and used phenomenologic classification of bacteria is that presented in the 9th edition of the Burgess Guide to Bacteria. In this edition, bacteria are divided into 4 or 3 main categories based on the structure of the boundary layer. The first, graciloculus, gram-negative eubacteria with the cell walls. Next, firmicutes, gram-positive eubacteria with cell wall. And next, tenoricutes, eubacteria devoid of cell walls. Archaea. Archaea is a group of single-celled prokaryotic organisms that have distinct molecular characteristics separating them from bacteria, as well as from eukaryotic cells. About characteristics, biochemical properties indicate that Archaea are independent group within prokaryotes and share similarities with both bacteria and eukaryotes. Major examples of these traits include the first about cell wall, the next about fatty acids, a third complexity of RNA polymerase, because archaea, like eukaryotic, have several RNA polymerases. And therefore, archaeal RNA polymerases are more reminiscent of eukaryotic RNA polymerases than bacteria. The next 4, protein synthesis. The various features of protein synthesis in the archaea are similar to those in eukaryotic cells, but not in bacteria. And archaea have an initiator or tRNA with unmodified methionine. And last, about metabolism, because methanogenesis, it's the production of methane as a byproduct of metabolism, occurs only in archaea, in particular in the Archaeotid subdivision. Classical photosynthesis using chlorophyll has not been found in any archaea. About reproduction, like bacteria, they reproduce by division. The process by which an individual cell reproduces is only chromosome and splits in two. About classification, archaea are divided into three polygenetic groups: Archaeota, Archaeota, and Archaeota. A first group consists of extremely thermophilic bacteria. Most of these metabolize sulfur, some of which reduce iron and molybdenum ions. Second group includes obligate anaerobic methanogenetic Archaebacteria, as well as extreme thermophile and holophase. Third group is formed by Archaebacteria living in hot sulfur springs. Virus is infectious agent of small size and simple composition that can multiply only in living cells of animals, plants, or bacteria. The name is from a Latin word meaning slimy liquid or poison. The earliest indicators of the biological nature of viruses came from studies in 1892 by the Russian scientist Dmitry Ivanovsky. All true viruses contain nucleic acid, either DNA or RNA, and protein. The nucleic acid encodes the genetic information unique for each virus. The infective extracellular form of a virus is called the virion. It contains at least one unique protein synthesized by a specific genus in the nucleic acid or that virus. In virtually all viruses, at least one of these proteins forms a shell around the nucleic acid. Certain viruses also have one proteins internal to the capsid, and some of these proteins act as enzymes, often during the synthesis of viral nucleic acid. About reproduction, viruses use two processes for replication: the lytic cycle and the lysogenetic cycle. Some viruses reproduce using both methods, while others only use the lytic cycle. In the lytic cycle, the virus attaches itself to the host cell and injects its DNA. Using the host cellular metabolism, viral DNA begins to replicate and form proteins. Then fully formed viruses are collected. These viruses destroy or lyse the cell and spread to other cells to continue the cycle. And about classification, viruses occupy a special taxonomic position. Shapes of viruses are predominantly of two kinds: rods or filaments, so-called because of the linear array of the nucleic acid and the protein subunits, and spheres, which are actually 20-sided polygons. Most plant viruses are small and are either filaments or polygons, as are many bacterial viruses. The largest and more complex bacteriophages, however, contain as their genetic information double-stranded DNA and combine both filamentous and polygonal shapes. Animal viruses exhibit extreme variation in size and shape. The smallest animal viruses belong to the families Parvoviridae and Prokaryotes and measure about 20 nanometer and about 30 nanometer in diameter, respectively. Viruses of these two families are isohydrons and contain nucleic acids with limited genetic information. Viruses of the family Poxeridae are about 2050 to 400 nanometer in their longest dimension, and they are neither polygons nor filaments. We have completed the study of Prokaryotes and eukaryotes, examined bacteria, Archaebacteria, and viruses. This is followed by a study of the classification of plants, their ecological forms and significance.